Hello everyone, this is MEENU AGRAWAL. So today I am going to talk about endomembranous system. As you know, this endomembranous system, endo, look at this word endo. Endo means what? Endo means within. So endo means within. So endo means within. So it's a system which is within now the cell, and it is connected, and here the one organelle is connected via membranous structure. Connections are made. Like how you can see here, the nucleus is there in the diagram. Okay, you can see this is the nuclear membrane is having a connection with your endoplasmic reticulum. Okay, and the whatever is being formed in the endoplasmic reticulum is just put into small vesicles, bag-like structure, and it comes and moves into the Golgi body. Okay, and Golgi body then does processing. We will see what it does, and then it makes out that complete product, and product comes from it, and then again it fuses with what? The membrane. So therefore this is called as what? Endomembranous system. So what are the organelles involved in it? What are the organelles if we talk about involved in endomembranous system? It's your nucleus, it's your endoplasmic reticulum, it's your Golgi body, your no, there's lysosomes. They are involved in it. Okay, and if we talk about the small vesicles, which are transport vesicles, you can say they are also in it. Transport vesicles, you can say, they are also involved in it. Okay, so basically if you look at somewhere, since we call it as an endomembranous system, so only these organelles are there. But if you look at the membrane is also now, the plasma membrane or the cell membrane is also a part of it, because plasma membrane is constantly being, now something is moving inside the plasma membrane and something is moving out of the plasma membrane, which is a part of this. Now you will see it's a part of your endomembranous system. So but we don't include plasma membrane because we are talking about within the cell. But remember that plasma membrane is also one of the major part of it. Okay, now moving on to the, first of all, it's the nucleus. So we will talk about the nucleus. So let's move on to the nucleus. As you can see, this is the three-dimensional structure I have put forward for you, so that you can have a good view about how the cell is. Cell is also a three-dimensional structure. I told you it's a balloon-like structure, you can imagine, and the cell is also like a ball-like. Nucleus is also what? A ball-like structure inside it. Okay, now if you open that ball, how it appears? So you can see here that what it has got this outer membrane, this outer membrane is there, and this one is your inner membrane. Okay, and this outer and inner membrane, they make around 10 nanometer. Now if I talk about, they make around 10 nanometer is the thickness of it. Okay, and this and in between, now you will see, look at the membrane, there are what? There are these small pores. If you can look at the diagram very carefully, you will see that it has got small pores on it. Okay, so these are nuclear pores. So together your outer membrane, inner membrane, and nuclear pore, they make up your what? Nuclear envelope. Okay, they make your nuclear envelope. And what do you see? That this is their nuclear envelope. Now if you look at nuclear pores, their size is around your 50 to 100 nanometer, you can say, not more than that. And see, substances just to the inner membrane, there are various basal lamina. We will study about cytoskeleton, which is holding, is present, which is holding your your chromatin. You can see your thread like structure, which is called as what? Chromatin. Okay, we will talk a lot about this chromatin. Okay, now if you look at these nuclear pores, through these nuclear pores, substances can move in and out of the cell in both the directions. Okay, it's not that key only in one direction, in both the directions. Like which substances will move inside it. Like smaller water soluble substances can easily move in and out of it, but bigger molecules like proteins which are being made outside in the cytoplasm, okay, now they want to move inside because they are needed in the nucleus. So when moving inside, it's highly controlled mechanism, okay. And when something like RNA and all, they are moving out of it for now you will read about key what is RNA and all. So RNA, when messenger RNA comes out from the nucleus out, then now and only the mature messenger RNA will come out of it. So various things are taken care of by the nucleus, okay. Nucleus is also known as what? It's known as brain of the cell. Why it is called as the brain of the cell? Because it controls the entire activities of the cell, okay. How it controls, what it has that controls, okay. So we will talk about all these things, okay? How they are controlling, what they have which is controlling, okay. So if you look at this structure, यहाँ पे आपको एक dense region दिख रहा है. You can see this is the dense region which we are calling it as what? Nucleolus. Now this is the region where your ribosomes are formed. So ribosomes, remember, they are involved in protein synthesis. They are involved in protein synthesis. So they help in protein synthesis. Okay. So nucleolus is the region, is a dense region which is basically involved in the formation of what? In the formation of your ribosomes. So they are involved in the formation of your this ribosomes. Okay. Now they are called as the brain of the cell I was talking about. So let's move on. As I was telling you that your cells have what? This nucleus is what? Is the brain of the cell, isn't it? Is the brain of cell I'm saying. So why we are calling it as brain of the cell? Brain, what does the brain do? Does it controls? It controls your body. So it controls all the cell activities. Now the question rises, what is there? What is there? Now, which is controlling? Okay, what is there which is controlling, and how is it controlling? There are two things. Okay, so now moving on with this, what is there? Now I showed you that structure, and inside it you saw there with what? Thread like structures which you call as chromatin. Now in that chromatin, if you look at, chromatin is made up of what? It is made up of your DNA and proteins. Now you know the DNA is what? DNA is, the full form of DNA is your deoxyribonucleic acid. Okay, so DNA, D nucleic and A. So DNA. Okay, so and it is made up of protein. So you will read about this molecule in detail in biomolecules, okay. But for this moment, just understand that this chromatin is made up of DNA and protein, and this DNA, why we are calling it as that it is a controlling factor? Because this DNA has got an information. What it has got? It has got an information, okay. And this information, now whatever information is there in it, like it has got the information that is passed on from your parents to you, like how will be your eyes, how will be your nose, what will be your skin color, all this information is there on this DNA. So this DNA has got all the information, when what type of proteins will be formed, when it will be formed, everything, they have got how, now what should be the structure of that protein, okay. Every kind of information they have, okay. So this DNA is what? It's basically that molecule which is controlling all the activities. Because why? Because it has the information. And how does it express that information? If I talk about that information, so that information, now it gets passed off from DNA to messenger RNA. Okay, and that process we call as transcription. I am repeating. See, you are reading it this term for the first time, so you are thinking, what is it? Just listen, just remember it once. So when many times it will go in your brain, it will seep into your brain automatically. Okay, so what is transcription, you will come to know. Okay, so when this deoxyribonucleic acid, this information is passed on to messenger RNA. This messenger RNA, you can see in the eukaryotic cell, there is a membranous separation of your genetic material and your cytoplasm. So in cytoplasm, what is happening? This messenger RNA from this nucleus, see this messenger RNA, from the nucleus, it is formed in the nucleus. Okay, it moves out into the cytosol, it moves out into the cytoplasm from that nuclear pore. Okay, so when this messenger RNA comes out from that nuclear pore, then what is happening? It is getting now translated. Translation happens here. It gets now that information is in a coded form. Okay, that information is read with the help of your what? This translation is done with the help of your ribosomes. So this is done with the help of your ribosomes. Okay, so this is done with the help of ribosomes. So messenger RNA, when it comes now out, then ribosomes attach on it. Okay, so just let me draw for you. Suppose this is the messenger RNA, this is the messenger RNA, I'm just drawing it here for you. This is the messenger RNA, and you will find that many ribosomes are attaching, and this like structure you must have seen. Okay, so they are forming what? They are forming your proteins. Okay, so this is called as polyribosome. I'm just introducing you terms right now, so that you become aware with these terms, so that when I will come to ribosomes, I will talk about it. Okay, so this ribosomes, they help in translation, help in formation of what? Proteins. So looking on that information which is there, which has come from DNA to messenger RNA, okay, and that information I told you is in a coded form, it gets decoded, and then your proteins are formed depending upon the message it has. It's the same analogy I can give you, like whatever information I am giving to you, you are going to write it in your examination. Okay, now if I give you correct information, you write the correct thing. If I give you the wrong information, you write the wrong thing. Same thing here happens. If this information on the DNA is correct, then what will happen? Your correct protein will be formed. The proteins formed will be correct. If this information is wrong, then the wrong protein will be formed, and therefore you find that there are genetic defects arising. Okay, we will talk about it later. Now there is a term which we call as, बहुत ही commonly used होता है, but अगर पूछा जाए तो बच्चों को पता ही नहीं होता है, but they keep on using this term, genes, है ना? What is a gene? What is a gene? Okay, what is a gene? Gene is अब ये भी मैं जो बता रही हूँ, ये भी पूरा तरीके से correct नहीं है. As you explore about it, you will come to know, because like for species we cannot give one particular definition. Similarly for genes we cannot give one particular definition. Okay, so but the most appropriate one is this I'm telling you. Okay, so genes is what? Gene is that segment of gene is what? It's a segment, you know, it's a segment of DNA. Is a functional, you can say it's a functional ONAL functional segment of DNA which codes for a particular protein. Okay, so what is gene? So gene is a functional segment of DNA which is coding for a protein. Okay, so that is called as what? Gene. Okay, so remember that. So the entire DNA is very long. Okay, but we are calling that part of that DNA which is coding for a protein. The entire DNA doesn't codes. Okay, so we will talk about it more when we move to cell division chapter about that, and you will read about DNA molecule in biomolecules chapter. So more and more things will become clear to you as you keep on moving. But right now remember that why we are saying that the now nucleus is the brain of the cell, because it is having all kind of information. कब क्या होगा, जैसे आपके ब्रेन को पता है कि कब क्या हो रहा है, कैसे ये हाथ मूव करना है, कितना मूव करना है, all these things is present now known to your brain. Similarly this is known to your cell. And why is it known? This is the thing. Now moving on, that you saw कि there is what? Chromatin. Okay, now you must have heard about this term also, which is called as what? It is called as chromosomes. So let me talk about chromosomes. Now if you look at this structure, okay, this is called as what? Chromosomes. Now the question arises ki how chromosomes are formed. The first question is how chromosomes are formed. How chromosomes are formed. This is one thing. Okay, and secondly is that ki when are they formed. There are two things. Okay, how chromosomes are formed and when they are formed. So we see them when they are formed only when the cell enters into the divisional phase. Okay, so you won't see them under the conditions when the cell is not dividing. Okay, so in a resting cell, in a not dividing cell, you won't see chromosomes. You will see them only in a dividing cell, first of all. Secondly, how chromosomes are formed. Okay, so see, chromosomes are made up of chromatin only. Okay, so chromatin, I again say chromatin condenses into rod-like structure called chromosomes. So chromosomes भी आपका किसका बना हुआ है? जाहिर सी बात है डीएनए और प्रोटीन्स का ही बना है and nothing else. Now if I say that the question is कि why are they condensing themselves? And if you look at now DNA, for your information I'm telling you, your DNA is only 2 nanometer. Okay, and this chromosome which I'm showing you is a metaphase chromosome. You will know about them later on when we will move to cell division chapter, and this is around 700 times condensed. Now why, first of all the question arises, why do they condense, and the second thing arises, how do they condense. How we will read later on, because more and more information about DNA is needed in that case. And why, the question is why it get condensed. The answer to it is that if something you have to transfer from one place to another, and that thing is very long, you know, if I give you suppose many sticks or bundles of many sticks I give you, okay, and I ask you to move it from one place to another place, or if I give you many books even so what do you basically do? You make bundles of it and then only you move it, no? Why you make the bundles of it? So that you make bundles of it so that no any part of it doesn't get skipped, है ना, उसका कोई टुकड़ा इधर उधर न हो जाए, गिर न जाए, कोई किताब गिर न जाए, कोई स्टिक गिर न जाए, इसलिए हम उसको बांधते. इसी तरह सेल्स भी एश्योर होना चाहती हैं कि दैट एंटायर जेनेटिक ना मटेरियल इज ट्रांसफर्ड, उसका कोई पार्ट टूट न जाए. अब बहुत बड़ा थ्रेड है, लंबा थ्रेड है, पतला थ्रेड है, तो बहुत पॉसिबिलिटी है कि इट विल ब्रेक. ओके, सो टू प्रिवेंट दैट, लाइक यू मस्ट हैव सीन, मैं एक एनालॉजी आपको देती हूँ यहाँ पे. आप सब लोग ने घर में रील का धागा देखा होगा, है ना, धागा का रील देखा है ना, कैसे कितना लंबा थ्रेड होता है उस रील पर उस छोटे से रील पर आ जाता है. क्यों? क्योंकि उसको लपेट दिया गया. तो समझ जाए कि कुछ ऐसे ही यहाँ पे भी किया गया है. ओके, So this, therefore it gives rise to this rod-like structure, which we call as what? Which we are calling it as chromosomes. ये आपका chromosomes है, okay? This is your rod-like structure, which we are calling it as what? As chromosomes. Okay? So ये पूरा आपका chromosomes आप देख पा रहे हैं. तो इसके different parts हैं chromosomes के. What are the different parts of the chromosome? This is your the primary constriction. जहाँ पर ये पहली बार जहाँ पे constricted region है, this is called as centromere. Okay? And at the centromere, you will find that it has got a proteinaceous covering. Okay, it has got a proteinaceous covering over it, and this proteinaceous covering is called as kinetochore. What do we call that covering as? We call that covering as kinetochore. So it has got a covering which we call as kinetochore, which now during cell division you will see that it helps in attachment of spindle fibers. So spindle fibers, they attach to this proteinaceous core. Okay, now if you look at the DNA, it has got a short arm and a long arm. Okay, and it has got at the ends of the DNA are called as what? They are called as telomeres. We call them as what? We call them as telomeres. Okay, now if you look at the DNA, there are what? Now you have got, if I talk of the two terms, I will talk of the two terms, that is haploid and diploid. It's very necessary to understand these terms. So you know that humans have how many chromosomes? We have 46 chromosomes. Okay, now means that is one set, that is 1 to 23 you have got from your mother. Okay, this is one set, and 1 to 23 you have got from your father. Okay, therefore you have got two set of chromosomes. Okay, or in an analogy way, if I tell you, if I write A, B, C, D till Z, okay, this is one set which is called as what? Haploid. Okay, and if I write attached to it, like if I write A, B, B, C, C, D, D till Z, Z, they become what? They all are occurring in pairs. So we call them as what? We call them as diploid. We call them as what? Diploid. Okay, so what do you find? That an organism has got, humans, like if I am taking, it has got diploid number of chromosomes. But your gametes basically have haploid number of chromosomes. For the time being, you just know this. Okay, we will talk more about haploid and diploid organisms later also. So आप जान जाइए कि haploid में there is only a single set of chromosomes. Okay, that's what I was saying. अगर 1 to 23 आपके ovum से आया है तो और 1 to 23 आपके sperm से आया है. ठीक, तो 1 to 23 is one set, like A to Z. हम जब बात करते हैं ABCD की तो वो एक entire set है ना. अगर हम उसमें कैपिटल और स्मॉल लेटर दोनों मिला दिए तो वो क्या हो गया? पेयर में हो गया, तो वो डिप्लॉयड हो गया. ठीक है, तो इसी तरह जब 1 क्रोमोसोम आ जाता है तो वी विल टॉक मोर अबाउट इट इन सेल डिवीजन. ओके, व्हाई आई एम टेलिंग यू हियर? बिकॉज हियर you see these chromosomes. Okay, you see these chromosomes are in pairs. Okay, and these chromosomes are called as these paired chromosomes, like 1 chromosome, 2 like A B B, and suppose this is from mother A and this is from father small a. Okay, so these paired chromosomes are called as what? Homologous chromosomes. So paired chromosomes are called as homologous chromosomes. Why do we call them as homologous chromosomes? Try to understand that. Okay, so we call them as homologous chromosome. Homo, look at the word homo. Homo means same. Logos means position, loci. It's a mathematical word. Okay, so what does it mean? That if I say here is your I gene, if I say, suppose I say this is your I gene, so in both the chromosomes, here will be what? I gene. Okay, fine. If I say here is your leg gene, okay, so in both the chromosomes it will be the same leg gene. Okay, so homo means same, logos means position. So if you look at homologous chromosomes, they are what? They are same in their what? In their shape, size and position of genes they carry. Same shape, size and position of gene they carry. Okay, so they are same in that. Okay, so okay, fine. So homologous chromosomes are such chromosomes. So you can see here that each chromosome is shown in pair. So they are homologous chromosomes. Okay. Now there are certain terms below it which you can see. Here is what? Metacentric. There are different types of chromosomes if you look at their shape. Okay. Depending upon their shape, chromosomes are now categorized further, classified further as metacentric when the centromere is in the middle. Okay, then it is called as metacentric. Submetacentric कब कहते हैं? जब वो एक तरफ थोड़ा सा shift हो जाता है. Then it is called as submetacentric. Okay, acrocentric means it is now more towards one of the arm. Okay, these are the two arms as you can see. Short arm is also called as P arm and the long arm is also called as Q arm. Okay, and when you move on you will see that it is moving towards one of the arm more. Okay, so one of the arm becomes more shorter, another arm becomes more longer. Okay, so short arm becomes even more shorter. So this is acrocentric. And telocentric, where you don't even find the short arm. Okay, quite at the ends, at the telo means ends. So at the telo position, at the end position, you will find what is there. At the terminal position you will find that the centromere has come. So depending upon their position of the centromere, it gives rise to different shape of the chromosomes, and that's how your chromosomes has been classified. Okay, so this is about your nucleus. Now okay, I have completed now your nucleus. Okay, with this I will say goodbye to you people, but keep logging yourself and keep subscribing me and keep watching, because in the next video I will deal with endomembranous system part 2. Okay, which will be even more interesting for you people. Okay, with that I say goodbye and God bless you all.